All right, well, thank you so much. I'm Jessica. I'm a PhD student in Dr. Sleiman's lab at McGill, and I'm really excited to talk to you about a couple stories today over the umbrella of reprogramming DNA with a small molecule. So just quickly, what I'll be going over today is the motivation for new strategies for DNA-based therapeutics, as well as an introduction into the world of these small molecule DNA hybrid materials. And then we'll talk about the very first one, cyanuric acid with polyadenine, its discovery and its application. And then I want to talk about the project that I'm working on, the newest class of these small molecule materials, with the small molecule being xanthine. So I also wanted to start with this quote because I think it sets the stage really nicely. And I think that something that nucleic acid chemists do really well is be inventive and be creative. And I really wanted to thank OTS for creating that space and also thank them for giving me the opportunity to engage and participate in it. Now, something I also love is this paradigm in science that structure informs function. And we're all familiar with this, so I won't belabor the point. I just find it really highlights nature's elegance, and we see examples of this everywhere, like the shape of our cells and organelles being the things that dictate their function within our body. And then we make these choices every day with the materials that we use. So of course, we all know that the elucidation of the structure of DNA then opened the stage for so many very interesting discoveries. And that's really where the stories today are kind of where they stand. We wanted to ask the question, can we explore a wider chemical space beyond just the four letters of DNA and investigate new structures with the use of using them as nucleic acid therapeutics? So of course, these projects aren't the first to address this. Seminal work was done by Paul Rethman in making DNA origami, and from that, we were able to use strategies that make discrete or large nanostructures in shapes and sizes that we get to dictate. Then we can also functionalize them to expand the scope of the chemical modifications beyond just the DNA code. But these do have their own limitations. It does require a lot of DNA in order to do these, and the functionalization and purification do have their own complexities. Now, each of these examples come from their own paper, but I did find them in one review, and I thought it was interesting. The authors of this review remarked that functionalization and the density of incorporation is something that's often overlooked and kind of limits the use of some of our techniques in terms of nucleic acid therapeutics, and that is something that these projects do touch on. And then, of course, we can make new structures of DNA by modifying the bases themselves, but these have their own limitations of usually being complex and laborious to do, which limits their greater accessibility to the scientific community. So the Sleiman lab wanted to answer this question of, can we add additional chemical handles with a high density of incorporation, but by doing so in a scalable and accessible way? So in order to address this, they're very inspired by the way that adenine hydrogen binds with thymine. And they noted that there's this small molecule, cyanuric acid, which has three thymine-like binding faces. And they proposed that it could potentially hydrogen bond with adenine, akin to the way that thymine does. Now, there's kind of two reasons why this wouldn't happen right off the bat. One, we're asking things to hydrogen bond in water, and also there could be a large entropic cost associated with bringing all of these free small molecules into one ordered structure. But kind of, in the same vein as that quote, motivated by curiosity, they tried it, and they actually saw that when cyanuric acid is mixed with strands of polyadenine, we actually see the appearance of this new structure, which is a triple helical structure. Just kind of take a moment to appreciate how cool this is. These small molecules are not intercalating and they're not groove binding. Instead, they actually directly participate in creating this hydrogen bonding motif, which then creates this triple helical structure. So here it kind of forms this helicene. And then they were able to characterize the structure on AFM, and they saw the assembly of long cooperative fibers. So to take this work a step further, Dr. de noted that you actually only needed 2 out of the 3 thymine-like faces in order to form the structure. So he created this library of alkyl chains and noted that subtle changes to these alkyl chains actually led to pretty significant differences in fibers. Now, from these various fibers, he then created DNA hydrogels, and they had the typical properties of being self-healing and stimuli responsive, but they actually showed record stiffness. And this is from unmodified DNA, and this is a really nice example of how we in fact have a new structure, that was able to create a material with unprecedented properties. Now, again, to understand this more and to exploit the very interesting properties of this material, Dr. Christophe Lachance-Spray worked with Sally, who's now currently leading these projects in our lab, and they noted that this triple helical structure is relatively resistant to degradation by nucleases. And so they wanted to know, can we confer this protection onto non-polyadenine sequences? So they wanted to protect these non-polyadenine sequences by kind of embedding them into this fiber. Now, they actually saw that the fiber conferred stability for over 18 days in the presence of cyanuric acid, whereas these ASO sequences, so these non-poly A strands, were only stable for less than 6 hours without that small molecule. So this is really interesting because it kind of showed this twofold, or they proposed this twofold protection mechanism. First being that the cyanuric acid poly A system is new to nature, and so the enzymes just have a difficult time recognizing and thereby degrading the DNA. And also, they leverage the fact that this is a supramolecular structure which forms hierarchically. So they propose that these non-poly A strands are actually kind of embedded and buried within this structure, thereby making them inaccessible to these enzymes. And then, as if this wasn't cool enough, they then showed a really applicable way to use this material in cell culture. So we're familiar with using DNA hydrogels in cell culture because they help cells differentiate, they have increased permeability of nutrients, but they have this central limitation of nuclease instability. So what they did is they used this material to encapsulate breast cancer cells, and then they actually saw their proliferation and aggregation. And this is really interesting because it shows promise that this new-to-nature structure enhances nuclease resistance and can be used in a day-to-day experimentation of cell culture, and could ameliorate studies with spheroids and maybe one day even organelles. And then as an interesting food for thought, these materials could potentially one day be used as like a reservoir for the prolonged release of nucleic acid therapeutics. And again, Sally is the one heading all of this now, and she's working with this technology with mRNA. So although this is really interesting and is really pushing the bounds of what we thought was possible, it also has its own limitations. So namely, kind of for the application in terms of anything biological, this system only assembles at pH below 7. And also, the critical concentration of the small molecule necessary is about 20 millimolar. So if there's a strategy that we could bring that pH to something more physiologically relevant and decrease that critical concentration, that'd be great. So that's kind of where we stand. Cyanuric acid is the first example of this new class of materials, the small molecule DNA hybrids. And then we have another example with melamine and polythymine, which is cool because it showed the extensibility of this approach beyond just one small molecule or one type of DNA. Now, melamine is interesting because it does have a more physiologically relevant pKa. However, it's toxic. So for anything therapeutic related, its application is pretty minimal. Now, I already told you the small molecule that I'll be talking about today. I like to refer to it as sitting in this very unbiasedly Goldilocks zone. But what I'll be speaking about for the rest of my presentation is the small molecule 3-methyl-xanthine. It has a pKa that we can work in physiological conditions, and also it actually has the lowest critical concentration of the 3 different small molecules. 3-methyl-xanthine is also a known therapeutic. It's used for asthma for bronchodilation. Now, this is interesting because it does expand the scope of application to more physiological relevant spaces, but also on a fundamental kind of structural point of view, this is the first example of a purine, and now we have 2 non-identical binding phases and only 1 thymine-like binding phases. So we can kind of try to learn more assembly rules of these new materials. So I use a lot of circular dichroism in order to kind of track structural changes. And here I'm showing you the CD signal for just polyadenine with 30 adenines on its own. And the whole thing is we want to know what's going to happen when we add this small molecule. And the idea is that changes to these CD signals actually are related to changes in the secondary structure, which could be related to an interaction between this small molecule and the polyadenine. So in fact, we do observe a significant change in CD signal upon the addition of the small molecule, which suggests interaction between the 2. Now we kind of took a step back and we noted that the parent molecule of 3-methyl-xanthine is xanthine, which is actually circulating within us right now. It's a degradation product of guanosine. And we started on this face capping experiment. So here the methyl's on the 3 position, but there's also 3 other derivatives of xanthine, whereby the methyl group cuts off either 1 or 2 potential binding faces. And we wanted to know which binding faces were necessary for structure formation with polyadenine. So what we did is we added each small molecule respectively to free polyadenine and took its CD signal. So here I'm showing you what we already saw, 3-methyl-xanthine. Now, if we add xanthine, we get something that's a little bit difficult to characterize. I don't go into it in this presentation, but would love to take questions on it. For now, we're just going to say it's a little bit of a question mark in terms of if the CD signal, the change is strong enough to suggest interaction. However, the other small molecule, 7-methyl-xanthine, deviates very little from the CD signal of the polyadenine on its own, so it's much more straightforward. There's likely little to no interaction. And the same can be true for theobromine, little change in CD signal, as well as theophylline. So we took a wider view at this, and we noted that actually 3-methyl-xanthine and xanthine are the only small molecules of this little library that have what we call the pseudo-Hugstein and pseudo-Franklin-Watson-Crick faces available. So we concluded that these 2 faces need to be open in order to bind with polyadenine and form a structure. So I keep referring to this structure, but what is the structure? Well, we wanted to know, we started off with an easiest motif, being a duplex. So we wanted to force the polyadenine strands to be anti-parallel to one another. And how we did this was by anchoring them to duplex DNA. So now we have this duplex DNA and the polyadenines are anti-parallel to one another, again, all in a duplex. And then we did the same thing, but for the parallel motif using reverse phosphoramides. So in black, we have just this anti-parallel pre-organized system on its own. In pink, we have the free polyadenine, so no pre-organization with the small molecule. And now we want to know what's going to happen when we add the small molecule to this pre-organized system. Well, we actually see 2 interesting things. First, that this pre-organized system really closely follows the CD signal of the free system, which suggests that the 3-methyl-xanthine polyadenine structure is predominantly in an anti-parallel duplex formation. Also, here we've corrected for the amount of DNA that we have, and intensity in CD can sometimes be related to order. And it would make sense that we have a more ordered structure with this pre-organized system because we're prepaying this entropic cost by already bringing the strands together. I also told you that we made this parallel motif. So here is the parallel CD signal on its own. And then in pink, we have the free 3-methyl-xanthine, which differs quite significantly from the parallel pre-organized system. Now, this is interesting because we actually saw that pre-organization can be used as a handle to leverage which structure we want to work with. So we understood that from a small molecule perspective, we needed the Franklin-Watson-Crick and pseudo-Hugstein faces. And now we understood that from a strand level, it was likely coming together in an anti-parallel duplex formation. And now we wanted to probe what was going on from the adenine perspective. So we're used to talking about DNA with the nucleobases in their anti-conformation, but of course they could also be in their syn conformation. So what we did is we constructed many different binding modes of how the adenines would interact with the small molecule, keeping in mind that there's variations in terms of the base orientation, the small molecule orientation, as well as the strand orientation. So I'm going to flash up all of the different modes that we were able to come up with, and it does feel a little overwhelming. That is how it felt for us. I'll add color just so we can kind of see the distinction with all those different variables in terms of the orientation of things. But really looking at this, the question is, how on earth were we going to determine the binding mode? Well, we turned to our collaborator and wanted to use molecular simulations to see if some of these binding modes were more energetically favorable. So I'll play the video if I can. Great. Oh, okay. sorry. So yes, so we turned to molecular simulations, and we actually saw that 2 out of the 5 binding modes were more energetically favorable. So we were inspired by the way that we did this base capping experiment with the small molecule. We kind of did the reverse of that with the adenine. Oh, okay. Hopefully, sorry. So yeah, so here's our adenine. And then what we did is we noted that if we add different amine groups, we can actually control the number of hydrogen bonds present. So here we made strands of 2 different derivatives with potential for more hydrogen bonds. And we thought that an increase in hydrogen bond could be tracked by using UV melting with a larger melting temperature. So we did this large experiment. I would love to take questions on it if anyone's interested. But the short of it is that from the molecular simulations and from our experiment, it looks like we're working with something in binding mode 1, where the adenines are in syn conformation and the strands are anti-parallel duplex formation. Then we wanted to characterize the system kind of as a whole, and we see potential fiber formation on AFM, which is cool because we could potentially leverage this for something like a hydrogel. But that said, these fibers are very different from the cyanuric acid fibers, where cyanuric acid forms these nice, long, abundant ones. These like to be in slightly more discrete tracks, which is also consistent with the DLS data that we have, which also shows something much more discrete. And then a big success of the cyanuric acid system was being able to create these derivatives on the face that's not participating in binding. And this system is also amenable to chemical modification, which is cool because we can further widen the scope of beyond just the functionalities of the regular bases. So in conclusion, I hope I was able to convince you that we did in fact discover a new motif in this small molecule DNA hybrid materials. We could impart structural control with pre-organization, and we see potential fiber formation on AFM, and the system is definitely amenable to chemical modification. Now, this is cool because we've expanded the scope of application into something more physiology relevant, but because this is the first example of a purine and has now 2 different binding phases, we could actually start to develop rules for assembly for these new class of materials. And then specifically for this 3-methyl-xanthine polyadenine system, we hypothesize that we're working with an anti-parallel duplex where the adenines are in the syn conformation. And current and future work is to really try to nail down the structure. So we're working with a collaboration to do solid state NMR, and then of course, further derivation to have some fun, cool, new small molecule derivatives. So thank you so much to Dr. Sleiman, to my mentors, and to the entire Sleiman lab for helping with all of this. And thank you so much for you guys for listening.
Thanks a lot for this interesting presentation. So if you have questions for Jessica, please use the Q&A function and type your question, and then we can use the chat or the Q&A to ask the questions. But I can always start with a question. I think during the presentation, you mentioned that this strategy is or can be used as a kind of a reservoir for a prolonged release, right? And I think so far the function is mostly in the mRNAs, uses mRNA release. So how easy is it, so what needs to be done to also make use of antisense oligonucleotides, siRNAs, or other applications? So what needs to be done, and how do you see the road forward?
That's a great question. And I'll just go to this slide. Really, Sally is the one heading this work in our lab. Let me, just grab here. What do I think? So how easy is it? This is a really interesting system because it's really easy to form. Sorry, I'm just trying to remember your question. It's very easy to form. And then what do I think that the road forward looks like? This paper is really nice. They talk about this ratio between the length of polyadenine and then the length of the strand that they're able to protect. So there is this interplay between needing filler strands of polyadenine. So they have had some very cool kind of ways around this limitation. But I do think that that's something that that Sally's currently working on in terms of the mRNA, just because it does have this really nice poly A tail, but it also has these non-poly A regions. So I guess to bury the lead a little bit, stay tuned for Sally's future work, because she will definitely answer those questions.
Yeah, and then maybe in the same line. So not sure whether it's part of your research, but can you also envision or think a little bit on the delivery, right? I think most of the work that you show is in silico or in vitro, but how to move forward to in vivo students or studies, or even to human beings.
Yes. So these hydrogels, these specific ones, so with the xanthine system, we're a little bit more in the fundamental stages, but with the cyanuric acid system, these hydrogels are injectable. So probably local delivery would be the best way to go for these. But again, the limitation of pH does mean that there's only certain areas in the body that these materials would be best suited for, um, which is a big motivation to kind of expand into that xanthine space. But I don't have answers to the xanthine questions just yet. Hopefully soon.
Okay, and I'm not the typical chemist, so I'm more a molecular biologist. So sorry if it's a very stupid question, but
No,
I
no.
think the limitation is the pH, right?
Yeah.
And what you showed in a later stage is that the pH of the three max, or how it's called,
Mhm.
had like a more workable pH. So how does that work actually? And why not? Is it not easier to adjust the pH of the previous compound?
It's a great question. It's a really good question. So here we need the nitrogens to be protonated in order to form the structure with polyadenine, but above the pKa, so like physiological conditions, they won't be. So they actually don't interact with polyadenine. Whereas here, the pKa is above that of physiological conditions, so we will have protonation, and it does in fact form uh, a structure with polyadenine.
Cool. Thanks. I think I give the word to Erin.
Yeah. I have a few questions that are more kind of technical in nature. Your stuff is really cool. Obviously, we're kind of more in the same space. And so I'm kind of interested in your characterization of the interactions by circular dichrosum. So I know that we use that tool a lot to investigate aptamers interacting with their targets. And we find that there are some challenges in the method itself, like the concentration has to be so high to be able, of the oligonucleotide, to be able to see any sort of like interaction. So do you find, this is like two questions, I guess. Do you find that limiting in your investigations? And do you have alternative methods that you use to investigate structure?
That's a great question. I love questions about CD. We do have a lot of different cuvettes. I don't know if that's something that you guys also explore. So like the different path lengths lets us kind of work at lower concentrations. So it hasn't been a limitation in this case. Also just working with polyadenine, like we can make that pretty easily. So
Yeah.
thankfully for us, it hasn't been a problem. I'm gonna knock on wood. And then the follow-up is what other kind of characterization techniques. I'm in the market for more. We have been trying to use DLS. It's not a perfect match because the system we don't think is spherical, but we are trying to work within the limits of that characterization. I do lots of UV-Vis melting temperature, AFM, and then I'm gonna be hopefully moving into like an NMR space. And I'm smiling now, but we'll see if I'm still smiling after.
A huge investigation. That's like, I
Yeah.
know in the past, people's entire PhDs have been on elucidating structure from NMR of oligonucleotides, right? So
Yeah.
good luck with the challenge. I'm sure
Thank
you're
you.
more than capable of taking it on.
Thank you.
So we have a great question in the chat, actually. I think anyone who's worked with oligonucleotides, worked with small molecules, knows that pH can be challenging, right? So they said, you mentioned that this motif only forms at a lower pH. What pH range does the original system work at? And have you seen it working in a wider pH range with your new system?
Yeah, so also a great question, and I think I'm on the correct slide. So we do just have this limitation of working above the pKa for the original system. So again, like delivery to places in the body that are more acidic is where the system really shines. in terms of like biological applications. It's got a lot of cool material applications. We haven't, so again, the 3-methyl-xanthine is newer, so we haven't like brought it into anything in vitro or in vivo yet, but just based on the pKa, we are excited about moving into a more physiologically relevant or more widely distributed area within the body.
Great. That's excellent. Last call for questions. It's a great talk. You know that there's always when there's fewer questions, it's because the talk usually is pretty good and explains things very thoroughly.
Thank you.
So, all right. If there's no more questions for now, you can still continue to ask questions of Jessica in the Q&A function or in the chat. She can answer them while Chris is presenting. But we're going to move to Chris. And thanks again, Jessica, for that great talk. And so whenever you're ready, Chris, you can take over.
Hi, can you see my slides?
Yes, perfectly. Go ahead.
So, hi, I'm Christopher. Thank you for attending today's webinar. And I'm also a PhD student in the Sleiman lab at McGill University. And today I will be presenting my research about enhancing Aptamer diversity for target therapeutics using DNA-encoded libraries. So let's start to talk about target therapy. So let's say we have a group of people all diagnosed with cancer. The conventional therapy would require the whole group to undergo the same time of treatment. However, target therapy would focus on specific biomarkers that are uniquely or overexpressed in the body of each patient and would target these biomarkers. And research have shown that target therapy can improve survival rates and reduce side effects when we compare it to a conventional therapy. So what has been done or what has been used as tools for target therapy? We have small molecules that can enter the cells and disrupt specific molecular pathways, specifically those involved in cell proliferation when we talk about cancer. And we have monoclonal antibodies that can go and bind to their specific target on the surface of the cancer cell. And we have for sure what we know as antibody-drug conjugate, where the antibody can bind to its target on the surface of the cell, and then the small molecules can enter the cell and cause the apoptosis. And we have the aptamers, which are emerging tools for target therapy. And their main function is similar to antibodies, where they can go and bind to their specific target on the surface of the cancer cell. So what are aptamers? So I like to compare them to antibodies. So we all know that antibodies are essentially proteins. So they start with the primary structure, which is a specific sequence of amino acids. And this primary structure can fall into secondary, tertiary, and quaternary structure to get our functional protein. In this case, it's our antibody that can now go and bind to its specific target. In a similar fashion, aptamers are short single-stranded DNA or RNA made of 4 nucleotides, A, T, C, G, or U in the case of RNA. And this single-stranded DNA or RNA can fold into secondary structure to get our functional aptamer, and this aptamer can now go and bind to its target. So if you can see, like they share similar functionalities. So why do we care about aptamer if that's the case? Because aptamers have higher shelf stability, can be regenerated, and they are quicker, less expensive to synthesize with minimal batch-to-batch variations. They have smaller size. However, the main, I would say the main limitation of aptamers is their poor or modest chemical diversity, and for sure the stability in the body and the presence of nucleases. But luckily enough, because the synthesis of aptamers can be fully automated, we can overcome some of these challenges by modifying them, which is not very simple in the case of antibodies or proteins. So what has been done in the literature to solve these limitations? So we have the work done by Somalogic, a biotech company based in the US, where people there have developed like slow of rate modified aptamers by using modified UTPs in their PCR, where they incorporate all these kind of modifications and they attached it at the C5 position of the uracil. So, however, the main limitation of this of this methodology is being always limited to one modification per library, and we're always limited by the polymerase active site. That's why I like also introduce the research done by the Hilly's lab at York University, where they use looper or ligase catalyzed oligonucleotide polymerization to derive their modified aptamers. And as you can see here, they overcame the problem of only one modification per library. However, we're still limited by the enzymes, either ligase or polymerase, and we're limited by the type and the position of the base where we want to introduce our modifications. Okay. So that's when Dr. Donatien had this idea of using non-nucleosidic modification to modify aptamers and to generate aptamer-like encoded oligomers, or what we call allenomers. So he started first with choosing a simple aptamer, which is the thrombin binding aptamer, or what we call TBA, and it's a G quadruplex. It's a short aptamer, 15-mer. So he decided to modify it. So he kept all the G's intact to keep the G quadruplex morphology, and he modified the other positions that are shown in colors here. And that's what I mean by a non-nucleosidic modification. You can see at position 6, we have a tryptophan modification directly coupled to a guanosine without any sugar, without any base. It's directly coupled to the guanosine using a small platform that we'll discuss afterwards. However, since we're using non-nucleosidic modification to be able to track this modification in future screening, we designed in this design, we have our aptamer arm and another arm that will serve as a barcode. So for each modification we would incorporate on the aptamer arm, we would have a specific coding for this modification. And for sure, to be able to have this branch structure, oligo structure, we need a branching unit, and that's the branching unit that Donatien synthesized in the lab. And for sure, we need the primer regions for any PCR needed in the future. So now I will be discussing the library synthesis strategy that will enable us to maximize the number of members per library. And that's all done on a fully automated solid phase DNA synthesizer. So let's say we started growing our branched oligo. Before introducing our first modification, we remove the CpG from the colon and we split it into 2 colons. And now we can introduce our first modification. On the first colon, we introduce the orange modification with its codon. On the second colon, we introduce the blue modification with its codon. And before introducing the second modification, we do this pool and split again. So we remove again the CpGs from the 2 colons, we mix them together, and then we divide them back and we put them back on the 2 colons. And then we introduce our second modification on each colon, as you can see here. So if we stop here, we ended up with 4 different compounds instead of having only 2. So it's 2 to the power n in this case. However, in Donatien's case, he was using not only 2 colons, he was using 6 colons. So it's 6 to the power, not 7, because he was introducing 7 modifications, if you can remember from the design that we introduced. So 6 to the power 7, it's approximately 300,000 members per library. For the selection process, it was done with the collaboration of the McGill lab at McGill University, specifically with Serge. And the selection process can be divided into 2 parts: a negative selection to eliminate any non-selective binding with the solid support and functionalized solid support. And then we collect everything that didn't bind in the negative selection, and we incubate it with our target immobilized on the support. And after a series of competitive elution with increasing the stringent conditions, and we do a final elution with heat, and we collect the best binders, and we PCR, and we send to next generation sequencing. And using this methodology, that's the top 5 sequencing sequence after next generation sequencing. You can see the first one is the TBA, the second one is allenomer A or modified aptamer A, and the third one modified aptamer B. You can see all the type of modifications in allenomer A and allenomer B. And these 2 allenomers have shown a better KD than the original aptamer without any modifications. So if you look at the graph D, we have like the binding affinities of ionomer A and B. The non-modified aptamer is not represented here, but it's like around 18 nanomolar in comparison to 10 nanomolar for ionomer A and 6 to ionomer B. So ionomer B showed 3 times improvement in the KD. And that's mainly because if you take a look at the graph B and C, which are the SPR results run, it's mainly due to a slower off rate for ionomer B, which is around 10-fold enhancement on the K off. So, however, the G quadruplex aptamer is a G quadruplex, and it's known to be thermodynamically stable, and it was a short aptamer. So to be sure that our methodology will be applicable to other aptamers, we need to validate it on a longer aptamer, which is a non-G quadruplex aptamer. So that's when Abdurrahman and Dr. Dani from our lab, with the collaboration of Dr. Iman from the McGill lab, started this project, and that's when I joined the lab and started working with them on this trastuzumab aptamer that you can see here. It's a 28-mer, it's a longer aptamer, and it's a stem and loop aptamer. And again, the modified positions are colored here. And using the same design, aptamer arm, barcode arm with the primary regions. And for sure, we still need the branching unit. Now it's a different branching unit just because it's commercially available and no need to synthesize it in the lab. And again, using this technology, we're able to select 3 better binders to trastuzumab. And the one at the middle with the histidine and T modification had the best KD. The KD was improved 5-fold. So what are these non-nucleosidic modifications that are key for this project? So we have mainly three groups. We have amino acid-inspired monomers, which are histidine, tryptophan, and phenylalanine. And usually we use these to mimic protein-protein interactions. And we have hydrophobic monomers, such as anthracene, naphthalene, dodecaine, to target the hydrophobic pockets of the proteins. And we have the alkyne monomer for any post-selection modifications. So how do we synthesize these monomers? So mainly we have 3 synthetic pathways in our lab. The first one and the most trivial one, I would say, is starting with diols. So we protect one hydroxyl group with the MT, and the second hydroxyl, we convert it into a phosphoramidite, and now we have our phosphoramidite that's ready to use on the synthesizer. The second synthetic pathway is using the tertiary amine carboxyl platform, and obviously this platform is used for peptide coupling to couple our amino acids. And as you can see, the platform is already DMT protected. After the coupling of the amino acid, we convert it into a phosphoramidite. And the third synthetic pathway is the tertiary amine alkyne platform. And obviously, this is used for any click chemistry. And then after we do the click chemistry, we convert it into a phosphoramidite. So what are the modifications that are synthesized using the diols synthetic pathway? We have the dodecaine or the C12 monomer. We start with the 112 dodecaine diol. We protect it with the MT. The yield is 37, but it's actually good because you always have a competition between monoprotection and double protection. And then we convert it into a phosphoramidite in dry solvent and under inert atmosphere. It's a very sensitive step to get our phosphoramidite. For the naphthalene, we start from the 2,7 naphthalene diol, but actually it's like a phenol, not a diol. So we still need to convert it into a diol form. So we reacted with the 6-bromo hexanol with the presence of base and catalyst. And under reflux for 48 hours, we get our diol form of naphthalene. We protect one hydroxyl with the MT, and the other one we convert it into a phosphoramidite. So that was for the diol synthetic pathway. So for the tertiary amine carboxyl platform, so how do we synthesize this platform in the first place? So to start with, we take tert-butyl glycinate hydrochloride, and we react it with 3-bromo-propanol in presence of base and catalyst. We get our diol form of our platform, and then we protect one hydroxyl group with the MT. And now we need to deprotect the tert-butyl group to be able to use it in peptide coupling. And we do this in basic conditions through hydrolysis. And then we add the tetrabutyl ammonium chloride to isolate our platform as a quaternary ammonium salt. So now we have our platform ready to use. So we start synthesizing our amino acid monomers. And here just disclaimer, we always use methyl ester protected amino acids to avoid any side reactions. So we start with our methyl ester phenylalanine hydrochloride. We couple it to our platform using peptide coupling in the presence of HATU as an activator. We get our coupled product, and then we just need to convert it into a phosphoramidite. Same thing for tryptophan. We'll start with the methyl ester tryptophan hydrochloride. We couple it to our platform, and then we convert it into a phosphoramidite. Same thing for histidine, but here we have one extra step, which is protecting the histidine with a trityl group. And that step is mainly done to increase the hydrophobicity of histidine, which is pretty polar. And for the ease of purification, it's preferable to protect it. So we start with the methyl ester histidine dihydrochloride. We deprotonate it, and then we react it with dichlorodimethyl and the silicon atom will coordinate to these two nitrogens, leaving the last nitrogen unprotected. And now we can add our trityl chloride to protect specifically that NH. And now we have our protected histidine, and we can do the same steps as before. So we reacted with our platform, we get our coupled product, and then we convert it into a phosphoramidite. And then this trityl group that we use to protect histidine will be cleaved at the end of the oligosynthesis. It's also acid cleavable like the MT. So the last platform is using the tertiary amine alkyne platform. So how do we synthesize this platform? We start from propargyl amine. We reacted with 3-bromopropanol in presence of base and catalyst, and we get our diol form of our platform. We protect one with the MT, one hydroxyl with the MT. And if we want to use it as a modification, we continue and do the phosphoramidite step. If not, we stop at the DMT step if we want to use it only as a platform. And like the case of anthracene. So for anthracene, we start with the chloromethyl anthracene. And with sodium azide, we add the azido group to it. And then now we can do a copper catalyzed click chemistry to get this product. And then we convert it into a phosphoramidite to get our final modifications. So for the future work, that was all about organic chemistry. So for future work, we plan to apply this technology or methodology towards more relevant aptamers that target kinase receptors. So I'm looking now at these two aptamers that binds HER2, which is a kinase receptor overexpressed mainly in cancer, in breast cancer patients. So I already synthesized these and purified these, so I still need to run a quick SPR test to check their KD and to see if I continue with these. And for sure, we're planning to include many more modifications. So one strategy is to synthesize dipeptide modifications. I've already started working with this, and I isolated this particular diphenylalanine amidite. And for sure, we're aiming for a way larger library. And we're also planning to include small molecule modifications. So let's say we work with HER2, that structure in red here. And the left is the lapatinib, which is an FDA approved drug for HER2. And for sure, it's better in the future to do our selection, not only using soluble proteins, but using cell models to really mimic the environment. So as a conclusion, this methodology was able to find a better binder for thrombin, for trastuzumab, and now we will be applying it towards target therapy. And the one cool thing about this technology is we're not always we don't have to always work with already published Aptamers, so we can design our own universal library and start like doing a high throughput screening towards many targets. So that was all for me, and thank you to Dr. Quentin, who trained me on organic synthesis when I joined the lab, and thanks to Abdelrahman also, and to all my colleagues and my friends in the Sleiman lab, and for sure to my PI, Dr. Hanadi. Thank you all.
Excellent. That was a great talk, Chris. It's always really cool to see the applications of this, because in the aptamer space, you know, we're always asked, can you rationally design an aptamer? Can you expand the ability of binding to things that are more like hydrophobic? Because we kind of always say the golden rule is if it can hydrogen bond, we can select an aptamer to it. But you're really showing the versatility of these molecules as molecular recognition elements, and kind of showing that it actually can be done, which is really exciting. And so I just had a quick clarification question. When you're talking about sequencing, how does do you have to do special conditions for the high throughput sequencing, or can you sequence with these modifications on? And how do you determine like which modification was in which place? I know you went through it briefly, but
So basically in the design,
Mhm.
why we have our barcode arm. So on the sequencing, so after PCR, you will not have any more the aptamer arm, right?
Oh, I see. Okay.
So you will decode this barcode to know which
Okay.
modifications we had. Yes.
Okay, perfect. I just wanted to make sure I understood that. It's so cool. It's like such an elegant solution. And so you've shown that you can apply it to better affinity for aptamers that are existing. Where do you really see the potential for these sequences to expand into different target regions to offer better affinity to over like traditional aptamers? Like, I
Yes.
guess the motivation. So you went over a little bit, but like, I think there's massive potential for these to expand. I'm just curious what your thoughts are on that.
Yeah. So basically, the two major things with these aptamers, for sure, is the binding affinity, which can be improved. So here, the improvement was not like crazy, because for trastuzumab and thrombin binding aptamer, they are already very good aptamers.
Yeah, exactly.
So modify it more, like it's not that ideal, but like we're aiming for, especially when we include our small molecules, because we've seen examples with 1,000-fold enhancement. So that's what we're aiming for. And if you can see like the stability is another good, because automatically you're introducing modification into your sequence, so it can increase the stability. So for example, here alanomer B, the half-life is 5 hours compared to 0.3 hours for the non-modified aptamers.
Yeah, that is so cool. That's awesome. I'm like, you know, this is a bit of a selfish invitation for me because I love the aptamer stuff, right? Also, it seems like you've done a ton of like, you know, nitty-gritty chemistry, right?
Yes.
And so you show a few slides with your synthetic pathways, but I'm assuming that's like months and months of work that has been
years of work. It was like before me and still optimizing till now. Yes.
absolutely. So do you think that it would be practical for another aptamer lab to kind of pick up this approach and be able to reproduce the chemistry, or do you foresee this being like a specialized company like SomaLogic? Like, what do you think is it generally applicable? Like, what are your thoughts there, just in the future?
Yeah. I think for an aptamer lab without any organic synthesis experience, it will be hard for the labs to adapt it. But what's happening now, like we got a fund, and which is like will be applied towards like developing a startup. Yeah.
Oh, that's great. That's really exciting. So
Another part of this project, where because the one limitation of this project is where to introduce our modifications, right?
yeah, of course.
So we have a new PhD student who's working on a machine learning platform
Oh, very cool.
to predict the modifications, and that's part of the startup. Yes.
I see we have some questions. I was hogging them all. Okay, so we have modification of aptamers using diverse libraries seems very interesting, but do you think introducing so many new modifications in a single aptamer can lengthen the journey of the aptamer from bench to bedside? As more rigorous studies would be required to study the pharmacokinetics and pharmacodynamics required, can this be a limitation of this process?
I'm not sure if I understood the question very well.
So I think to rephrase it, basically, you know, it's a long pathway for a naked aptamer that's unmodified to get
Yes.
regulatory approval. So having even more of these modifications, do you anticipate that it would like lengthen that pipeline so that there would need to be even more exhaustive studies done for safety profiles for these sequences?
I don't think that when they do these studies, they will do like a one by one.
Okay.
Yeah.
Okay, very cool. And then the last question before we wrap up is, which polymerase are you using to amplify the modified bases?
Okay, so again, because the main objective of this research is to overcome the limitation of polymerase while leading modification. So that's why here you don't need any special modifications again, or any special polymerases again, because of the design. We have the barcode, and we're reading this barcode, which is only regular nucleotides. So we're not reading the modifications.
Great.
Can I ask a question?
Oh yeah, sure. Go ahead, Ronald.
That's right. So, nice presentation, Christopher. So I have a question about the different kind of aptamers. So I think you have the stem loop aptamers, and you also showed G quadruplex aptamers, right? So I can kind of envision that the G quadruplex aptamers are way more stable than the stem loop one. So do you have any data on that?
So that's why we work with the trastuzumab aptamer as a next step, because the G quadruplex is very stable and it was short aptamer. So the methodology, okay, one can say it's easy to apply it only to TBA, but not to other aptamers. But we confirmed that this using the trastuzumab aptamer, modifying the trastuzumab aptamer, which is a 28-mer, a stem and loop aptamer. Yeah, so I don't know if I answered your question.
More or less. And I also have a question about, because you did the trastuzumab
Yes.
aptamers optimization,
Yes.
where you show that you, with your kind of unbiased library system, you could generate aptamers that are way more potent than the already existing one, right? So did you do any assays like a stability assay, a toxicity assay? Did you look at biodistribution, those kind of things? I'm
Not,
just curious whether these improved compounds are also less toxic and
no, not yet, not yet. So that was the TBA and trastuzumab were only like the proof of concept of this technology. So to make sure that this technology is working, and for sure the next steps would be incorporating such assays and the future. Yeah.
cool. Yeah, thanks a lot.
So thanks again, Chris. It was an awesome talk. It's really exciting to see the chemistry side of things from McGill. That's always fun. And so I just want to remind everyone that we do have some webinars coming up. So on March 27th, we have a webinar to meet the new trainee reps for the board of directors of the OTS. So that should be good. And then on April 10th, we have another trainee spotlight series. So we're excited to welcome you back for that.
Oh.
And if you, sorry, go ahead, Ronald.
I just see 2 new questions in the Q&A.
Oh.
Do we have 2 minutes to answer the questions?
Yeah. Okay. So, is there any possibility that the single-stranded DNA can influence the structure of the aptamer?
So with modification, because
Yeah, it must be the modification to the.
Yeah, maybe the modifications. So yeah, for the atomic binding aptamer, we have like experiments where we check the G-quadruplex structure with the modification. So, and the G-quadruplex structure was preserved using the modifications, yes.
Great. And it seems like a lot of the questions that we've had for your work specifically are really like technical about the method. Is there a paper already published for this that you can direct us to in terms of like the specific details of how the selection works and
So for the thrombin binding aptamer, we have it on the CAM archive, and it
okay,
will be published like soon, the thrombin binding aptamer and the trastuzumab aptamer very soon. Yeah.
perfect.
Yeah.
Perfect. All right, so I think that will answer the questions. And like I was saying, we have a few webinars coming up, so keep your eye on the webinar page for the Oligonucleotide Therapeutics Society. And I just want to thank Chris and Jessica again for giving us great talks today.
Thank you.